So our next speaker is Koki Tsuyuzaki. He is a researcher at RIKEN in Japan. And he is also one of the organizer of this BioSeq Asia 2021 meeting. And he is an author of multiple bioconductor packages. Koki's latest bioconductor package is delayed Tensor, and this package has been added to the latest bioconductor release 3.14 version. So let's welcome Koki.
Thank you for introducing me today. Thank you for having me. I'm going to talk about my packages to deal with large-scale and complex sensor data in R. Today's talk is a little technical, and biologists might think it's a little boring, but I believe that it's very fundamental and important issue in any life science field. So I hope you are interested in my talk. First of all, I explain what is tensor. Briefly speaking, a tensor can be considered as a generalization of data representation. For example, a scalar value is tensor. This is called zeroth order tensor. A vector is also tensor. This is called first order tensor. A matrix is tensor. This is second order tensor. If a data has three modes, I mean height, width, and depth, this is called third order tensor. So that's why any data is basically a tensor, but in most cases, the term tensor implies the third order or much higher order tensor. Next, I will introduce the tensor decomposition algorithm. At first, I will explain the matrix decomposition, which is a special case of tensor decomposition. Let's say we have a matrix having gene times cell, and this matrix has some blocks having elements containing the bigger or smaller values. In matrix decomposition algorithm, this matrix is approximated by a small number of patterns, such as x1, x2, and x3. Each pattern can be represented by the outer product of two vectors like this. The first row vector has larger or smaller values here, and these are originated from this blue block. Likewise, the first column vector has larger or smaller value here, and these are originated from this blue block. We can normalize these vectors. After normalization, a scalar value can be extracted from these vectors. After setting these scalar values in the diagonal elements of this matrix lambda, we can formalize this decomposition as a product of three matrices like U lambda V transpose. Matrix V has three column vectors here, and each vector can be considered the set-wise pattern and can be used for visualization or as a set-wise analysis like the clustering. Likewise, this matrix U has three row vectors, and each vector can be considered the bin-wise pattern. The small diagonal matrix lambda can be considered the magnitude of each pattern. Since the tensor decomposition is the extension of the matrix decomposition, let's consider it in the same way. If we have a third order tensor, for example, gene times tissue times condition, using tensor decomposition, we can extract a small number of patterns. Each vector can be summarized to the multiple matrices, and these are called factor matrices. The scalar values are also summarized to a small tensor, and this is called core tensor. There are two types of tensor decomposition, CP decomposition and Tucker decomposition. In CP decomposition, the core tensor is assumed to be diagonal, which means we have to set the number of row dimensions of all directions are the same, which means J1 equals J2 equals J3. And also the relationship of vectors of different factor matrices are 1 to 1. And on the other hand, in Tucker decomposition, the core tensor is assumed to be dense, which means J1, J2, and J3 can be different. And the relationship of vectors of different factor matrices are many to many. Here, I show a use case of tensor decomposition. This is a TSME map of 1 million mouse brains single-cell MeSeq data. In this map, the cells are mapped in a lower dimensional space and have different colored cell types. And many single-cell studies have been detected cell-cell interaction or CCI between cell types on the basis of ligand-receptor co-expression. Ligand and receptor expression can be extracted as raw vectors from SR-MeSeq data. So we first calculate the outer product with these vectors. By the outer product, the order of a vector increases and become a matrix. And this matrix can be considered as a similarity matrix with all the possible combinations between the cell types. We can apply the outer product to many ligand receptor pairs. So the piled up these matrices become a third order tensor. In my study, I call this CCI tensor. Then I perform a tensor decomposition algorithm called non-negative Tucker decomposition against the CCI tensor. By this decomposition, we can see the representative triadic relationships in the CCI tensor. That is, we can see the correspondence between ligand expressing cell types, receptor expressing cell types, and the related ligand receptor pairs. Using scTensor, we could extract the triadic relationship, as you can see in this figure. Furthermore, our simulation dataset shows that scTensor has the ability to selectively detect only the correct CCIs compared to the label permutation test, which is commonly used conventional methods to detect CCI. This tendency is more pronounced when the CCI is many to many rather than 1 to 1. And it suggests that scTensor is an effective method for SR-MeSeq data containing more complex CCI. Tensor and tensor decomposition are very versatile and actually used for all kinds of life science programs other than CCI detection. However, there are still technical challenges to overcome. In this plot, these are all my R packages, and x-axis means how many tensors can be used in each package. It is also interpreted as the complexity of data structure. Y-axis means how many elements of tensors can be used in each package. And it is interpreted as the complexity of data size. Generally speaking, the larger the data size and the larger the number of tensors, and the more difficult it is to model such data. So some kind of framework to support such modeling is needed. Here, I will discuss the complexity of data size first. As the number of orders of tensor increases, the data size is getting bigger. For example, multiple third-order tensors become a fourth-order tensor, and multiple fourth-order tensors become a fifth-order tensor, and so on. That's why we have to think about some techniques to address this problem. In our previous benchmark study about principal component analysis, we found that out-of-core and sparseness are very effective for the scalability to analyze large-scale matrix. So I will extend these techniques to tensor decomposition. So now I am developing out-of-core and sparse implementation of tensor called delayed tensor. In this package, a tensor data is stored in HDF5 files, and the block tensor is incrementally loaded in an out-of-core manner, only when the data is needed. After data loading, each loaded block tensor is optionally formatted as a sparse tensor. This feature is achieved by using another package, a delayed array. In delayed tensor, some functions of RTensor and INSAM packages are re-implemented for delayed array framework. RTensor provides some tensor-specific arithmetic and tensor decomposition algorithms, and INSAM merges multiple tensors into a tensor as the syntactic sugar of 4 statements. Using this delayed tensor, we will be able to perform many tensors arithmetic and tensor decomposition, even if the tensor data is extremely large. This is a benchmark of delayed tensor. The red bar means the result of our tensor with our standard array. The green bar shows the results with dense mode delayed array, and the blue bar shows the result of sparse mode delayed tensor. And these left two plots are the memory usage, and the right two plots are the calculation time. Here, I tried the folding operation and tensor canonical product. And these left two plots show that delayed tensor can manage memory usage no matter how large the data size becomes. However, as you can see from the two plots on the right, the calculation time is relatively slow compared with our tensor, which means that the out-of-core implementation has a bottleneck of loading data from a file, which may take extra computation time. Actually, the calculation time depends on the block size to use each calculation. By setting the value, the users can control the trade-off between the memory usage and the calculation time. Okay, next, I will discuss the complexity of data structure. Biological data is becoming more and more complicated. And when we consider single-cell MeSHDbi data, which measures the amount of gene expression in each cell, various data are related to it, such as different omics data of the same cells, spatial coordinates, and so on. Likewise, gene-related information such as pathway database, the gene regulatory network, and the target gene's function are available. There are some open questions. For example, how can we represent such heterogeneous data? What kind of algorithms are available? Can we deal with such data in common framework? Tensor decomposition in complex structures is generally difficult to model or implement. Many papers propose decomposition methods for specific tensor structures like this, but such methods cannot deal with structure change in input data. And some methodologies can be applied to any tensor data structure if needed. To be able to decompose data in a unified way, no matter how complex it becomes, I developed MuTensor, which includes the most of existing tensor decomposition methods as special cases. Here, let's assume that we have three tensors, x1, x2, and x3. And x1 and x2 have a common dimension, I2. And x2 and x3 have a common dimension, I4. In MuTensor, the parameters for decomposing them, such as the number of dimensions and the decomposition algorithms, are defined as a parameter object. After that, the decomposition is performed in a unified manner by the function coupled MWCA. And here is the decomposed form of these tensors. What is important here is that tensors with a common dimension have a common factor matrix. In other words, this common factor matrix can be estimated by multiple tensors. For example, this A2 is estimated from x1 and x2, and A4 is estimated from x2 and x3. This assumption has the effect of extracting only the patterns reproduced by the multiple tensors as compared to decomposing the tensors individually. In order to demonstrate MuTensor, I will now explain something called a tensor network. So far, tensors have been explained as cubes, but these can also be represented as graphs using the tensor network notation. For example, a third-order tensor X with dimension I1, I2, and I3 can be represented as a graph with 3 legs in a tensor network. Now, suppose there is another tensor Y that has a common dimension I1 with tensor X. In this case, the tensor network is represented as X and Y being connected by a common leg I1. Here, I will demonstrate MuTensor. In order to show that it can handle extremely complex data structures, I prepared a dataset like this. Within this tensor network, there is a fourth-order tensor in the center. There are third-order tensors surrounding it. And there are second-order tensors outside the third-order tensors. Only the block diagonal elements are assigned larger values in this way, so that the patterns in the data can be visually checked. And here is the result of applying MuTensor to this simulation data. And these data sets are reconstructed from the result of MuTensor. And as you can see, the pattern of the original data can be accurately extracted. Now let's add random noise to only X3. Interestingly, when we look at the reconstructed data here, we can see that we have extracted only the original pattern and not the noise. This is because X3 is connected to the surrounding X1, X8, and X9. We assume that the factor matrices consist of X3 is common to these tensors. The same noise is not included in X1, X8, and X9 because it was randomly added only to X3, and it is not reproducible among the data, making it hard to be selected. I cut all the tensor legs between the tensors and decompose them individually. This revealed that the noise was detected in X3 like this. And moreover, the parameter estimation became unstable in other tensors as well. Thus, assuming common patterns among multiple data and decomposing them at the same time shows that the modeling of a mu tensor can be robust to noise. Okay, today I introduced these three packages: scTensor, decompose SGN-seq data, and detect CCIs. Delayed tensor enables users to use large-scale tensor data in out-of-core and sparse way. Mu tensor enables users to model complex tensor data. Please also check today's Japanese workshop to demonstrate these packages later. This is all for my presentation. Thank you for listening. If you have any questions or comments, please ask.
Thank you, Koki, for the very interesting talk.
Thank you.
Yeah, so just to remind everyone, so if you're interested in learning these packages, you can come to the workshops today. And the Japanese workshop today will be presented by Koki, and then he will introduce the packages developed by him. Yeah, so there is a question from YH Taguchi. It's about what would you like to minimize to get decomposition?
Minimize. Maybe he's talking about the okay. Minimize. Yes. Maybe he's talking about mu tensor. If it is the case, how can I say? Mu tensor. In mu tensor, users can select the algorithm to decompose tensor. So mu tensor has no specific model to decompose tensor. It's all of such setting is specified in a parameter object. Oh, he said, I see.
Thank you. Luke Gandalf asked, do you recommend any reference for learning more about the theory of tensors in data analysis?
Yeah, there are many good books or papers, especially non-negative tensor decomposition or matrix decomposition written by Dr. Amano. Dr. Amadi. It's very famous book and very sophisticated. So please check this book, please.
Thank you. And Matt Ritchie has asked, Regarding the fitting algorithms you mentioned, CP versus Tucker, your single cell example used Tucker. Are there times when CP would be preferred for biological data?
Yeah, of course. There are pros and cons between these methods. This is I can say CP has only a single parameter called rank, so it is easy to tune up the hyperparameter. On the other hand, Tucker has many parameters, so sometimes it is very difficult to tune such parameters, especially the data is very complicated. So I cannot say which is the better method in any data. And then in scTensor, I selected Tucker form decomposition. And this is because CCI tensor is not so complicated. And when I tried CP and Tucker, the result of Tucker is a little performed. So that's why I selected Tucker form tensor decomposition.
Thanks, Koki. So if other people have other questions, you can continue discuss in Slack. And we'll have a 3 minutes break, and we will welcome the next speaker, Luke.